Welcome to the student webinar. This is a Summer School edition 2025 edition. And that is all the students that won the Nobel Prize, I would say. Maybe it's a future, you have a good chance for this, but are all the students that won the poster prize at the summer school driven by BioExcel this year in June in Sardinia. And in particular, we have Jan for the National Institute of Chemistry, Slovenia, Gesa from the Max Planck Institute for Biology, Germany, and Susanna from the Institute of Organic Chemistry, Biochemistry, Czech Republic. I'm Alessandra, and I'm chair this webinar together with Otto and with Richard and we are all from BioExcel, the Center of Excellence for Computational and Biomolecular Research. So the webinar is recorded. And during the webinar, you have the possibility to ask questions using the Zoom button that you see at the end of the Zoom application. You could have this one or this one, depends on which operating system you have. There you can type your question. It will be great if you have a question for Jan, put at 1 or at 2 for Gesa and at 3 for Susanna. You will be great if you tell us also if you have a microphone or not, because now you will listen all the 3 presentations, and at the end of the 3rd presentation, we can unmute you to ask your question. Otherwise, I will read, or Otto will read the question for the speaker. So in particular, what will be the topic? Jan will speak about investigating catalytic inhibition of a full-length topoisomerase 2A through all-atom molecular simulation, while Gesa will speak about molecular dynamic simulation of AF1503, a model protein for transmembrane signaling. And at the end, we will have Susanna that will speak about oligoarginine peptides bind preferentially to concave phospholipid membrane and induce membrane multilamellarity. They are all interesting topics, so I'm looking forward to hear what you have to tell us.
Hi, my name is Jan. I come from the National Institute of Chemistry in Ljubljana, as Alessandra has said. And today I will be talking about our ongoing investigation of topoisomerase 2A, specifically its catalytic inhibition. So type 2A topoisomerases are complex molecular motors that alter the topological state of DNA chains. They act in processes such as transcription, DNA replication, and cell division, where they resolve issues like positive supercoiling, the formation of precatenanes, and interlinked sister chromatids. And by doing this, they are essential for the normal progression of the replicative mechanisms of the cell. They function in a complex catalytic cycle, which goes something like this. So first, the open topoisomerase captures a G segment, which is bound into the DNA gate. Next, a T segment follows, and after the capture of 2 ATP molecules, it is enclosed by the closing of the N gate. In the next stage, this G segment is cleaved to allow for the passage of the T segment, which is mediated through ATP hydrolysis. And lastly, this G segment religates or rejoins, and then the cycle through some additional steps reprimes and goes again. So this happens numerous times in our cells, in all of our cells, but particularly in cancer cells, which divide more rapidly and also indefinitely. So they are more affected. So they are more affected if we were to inhibit this cycle. And there exist drugs that do this. So these are called Topo II poisons. Etoposide is the main representative, which bind in this stage of the catalytic cycle. So when the G segment is bound, they kind of bind here to prevent the rejoining of the G segment. And by doing this, they cause an accumulation of double-strand breaks and increase the risk of secondary cancers. So in recent years, different drugs have become more interesting as a research alternative. These are ATP competitive catalytic inhibitors that bind before the cleavage of the G segment, and they bind into ATP binding sites here when the T segment is already captured. Specifically, we deal with these two. This is an in-house compound called BT. It is a bithiazole, and it has a quite high micromolar IC50. And on the other hand, we also have a Novartis-developed compound called QAP1 with about 100 times greater potency. So we are interested in how the binding of catalytic inhibitors alters what happens in this stage that makes it not progress to the next stage of the catalytic cycle. So our goal is to understand the mechanism of ATP competitive catalytic inhibition. And to do this, we use biomolecular simulations and combine it with different trajectory analysis tools to answer our questions. Before going into our study, let's just look at some structural details that will be important for the understanding. So this is the yeast type 2 topoisomerase from the front and from the side. We can see that it is a homodimer, meaning that it is made up of two identical chains, which make up an ATP-ase domain here at the top, which houses the T segment, and the DNA binding domain here at the bottom, which houses the G segment. Completely at the top are GHKL subdomains, which are the N gate that I was speaking about, that closes after the capture of the T segment. Then here in the interface, we can see K loops. These interact with either the G segment or the T segment. And lastly, here below the G segment, we see two catalytic tyrosine residues, which are the active site and are responsible for cleaving the G segment. As I said, we would like to simulate structures like this, ones that have the G segment and the T segment already inserted. But if we look through the PDB database, we see that those are not available. Specifically, the T segment has not been resolved experimentally. So my predecessors in the lab have used the full-length yeast Topo II. Here I only show the upper part, and performed targeted MD to pry open this upper N gate and then manually inserted the T segment into it. What we then did in this study is symmetrically into both ATP binding sites, we added either ATP, which is the native ligand that should progress the catalytic cycle further, or catalytic inhibitors, which should stop the progression of the catalytic cycle. And then we performed biomolecular simulations using AMBER, where we got about a microsecond of dynamics per system, plus some additional shorter replicas. So we want to understand how the protein dynamics differ in the presence of ATP, which is the native ligand, versus the catalytic inhibitors. And one of the most important things that could change is how the T segment is affected. So let's first look at this. To evaluate what happens to the T segment, we perform two different analyses. Firstly, hydrogen bonding analysis, which tells us in what percentage of frames are hydrogen bonds formed between the protein and the DNA. And on the other hand, we also perform an energetic analysis, which tells us what the free energy of binding is between this T segment and the surrounding protein, decomposed by each nucleotide of the DNA. And what we can see is that no matter the ligation state, the energy contributions are always unfavorable. They're positive, which also coincides with the fact that there are very few hydrogen bonds formed, especially if we compare this T segment with the G segment, which has many more. So this tells us that the T segment absolutely hates being here. It doesn't want to stay in the ATPase domain, but what does it want to do? So we have a few options. In the presence of ATP, we know that the G segment cleaves and the T segment is passed downward through the cleaved G segment. Whereas in the presence of catalytic inhibitors, we are not actually sure what happens. One possibility would be that the T segment exits the ATPase domain upwards through the N gate where it came from, or perhaps it stays bound here and maybe induces a conformational change. So let's look first at this option of the T segment leaving upwards through the open N gate. To to evaluate this, we look at the distance between the GHKL domains. So this is the AND gate, basically, as we have defined it. And we see that in the presence of catalytic inhibitors, this distance is reduced by about 10%. This means that catalytic inhibitors induce AND gate clamping, whereas ATP does not. And additionally, if we look at local PCA, we can see that not only is the extent of the motions different, also the motions themselves differ. In the presence of catalytic inhibitors, we can see that there is a sort of sliding motion of the GHKL domains here from the top. And in the presence of ATP, it is a twisting/sliding motion. So we now know that the T segment cannot leave upwards. So what does it do then in the presence of catalytic inhibitors? Well, to answer this, let's first look at what we would expect to happen in the catalytic cycle when ATP is present. Here I show the interface between the upper ATPase domain and the lower DNA binding domain. And because you probably don't see anything, I also show the human homologous Topo II alpha enzyme, where you can see that there is a crossover of protein chains happening. This means that if there was a T segment here, it is mechanically prevented from moving downwards by this obstruction here at the interface. So what would have to happen is some sort of counterclockwise twisting, which would of the ATPase domain up here, which would enable the passage of the T segment through the G segment. So now if we go back to the yeast system and look closer at this interface, we can see that also this twisting is not possible because there is an interaction between the G segment and one of the K loops, which you I hope remember from the structural presentation. So if we now take a step back, you can see that this interaction between the K loops and the G segment is caused by a global tilting motion of the ATPase domain, which engages the K loop on the G segment and prevents twisting. So if you go in the opposite direction, we would see that for the T segment to pass downward, we would have to first untilt the structure that would disengage the K loop from the G segment, allow for twisting, and then the passage of the T segment downward. But what do we see in our simulations? So first, let's look at the interaction between the K loops and the G segment. This is again an MMGBSA analysis where we now look at the free energy of binding between the K loops and the G segment and decompose it per residue of the K loop. What we can see is that the pink K loop does not interact with the G segment at all, whereas the purple one does, and quite strongly so. And additionally, no matter the ligation state, these interactions are about equally strong in all states. So would this mean that also the global motions, so that twisting and the tilting, are quite similar? Well, we measure those, and here we define tilting as the angle between the DNA binding domain and the ATPase domain, specifically their vertical axis, and twisting as basically the angle between the same domains, but then in the horizontal plane. And what we can see is that ATP-bound structures are more tilted and less twisted than catalytic inhibitor-bound structures. So if we take a step back, we said that we would expect that in the presence of ATP, the structure untilts and twists. So untilts and twists. But we can see that, in fact, catalytic inhibitor-bound structures are more likely to do so than ATP-bound structures. So how do we explain this? In the presence of ATP, we expected something like this. So first an untilting followed by a twisting, and then an ATP-mediated passage of the T segment. But we don't see this. What we propose happens instead is that upon the binding of ATP to the ATP binding site, an allosteric signaling pathway is activated between the allosteric site and the active site here, where the G segment is cleaved. And in fact, first, the G segment cleavage happens before the rearrangement of the ATPase domain. On the other hand, in the presence of catalytic inhibitors, We presume that this signal is not passed because the G segment isn't ever cleaved. Instead, we propose that maybe the T segment leaves upward to the N gate through the N gate, but we also saw that this is not possible because the N gate is clamped. And additionally, a twisting and an untilting motion occurs to trap the T segment in a sort of metastable state. So to recap, we set out to research how catalytic inhibitors alter protein dynamics. And we have shown that they induce a clamping of the N gate and then also large-scale motions called twisting and tilting to lock the T segment in a sort of metastable state in which it is unhappy but unable to proceed upwards or downwards. What we didn't set out to research, but kind of stumbled upon also, is what happens in the presence of ATP. So we propose that before this structural rearrangement, the ATP sends a signal down an allosteric pathway to the orthosteric site to first induce a G segment cleavage and then lead to this rearrangement of the ATPase domain. So in the future, we would like to further look into allosteric pathways. We have already started doing this. That's why I can show you one of these plots. And additionally, we would also like to simulate structures where the G segment is cleaved. Since then, we could prove if we can see all of these rearrangements that we have hypothesized about now. That's all from my side. Thank you for your attention. And I guess if you would like to ask some questions, you have to write in the comments.
Please, Jason.
Thank you. So you see my screen and hear me, right?
Yes.
Okay.
Yes.
Perfect. So let's start. So good afternoon, everyone. I'm Gesa Freimann, and I'm a first-year PhD student with Andrei Lupas at the Max Planck Institute for Biology in Tübingen. And when I started studying biology, I really wanted to know actually what drives life, what makes us alive. And this has fascinated me through biology, biophysics, bioinformatics. And starting the PhD, I was thinking, what is it that we understand least with regard to proteins? And I really think it's their dynamics. The sequence we can understand quite well. The structure now, especially with AlphaFold, we have a good idea. But the dynamics, I think, are well less explored. And especially the dynamics of transmembrane signaling proteins. And this led me to the PhD project of doing molecular dynamic simulations of A1503, which is a model protein for transmembrane signaling. And two-component signal transduction systems are there in bacteria and archaea to detect various stimuli. So these two-component signal transduction systems composed of a first component, as the name says, which is a histidine kinase that detects a signal, transmits the information across the membrane, and then phosphorylates, which then triggers the phosphorylation of a response regulator, which then triggers a cellular response. And the signals can be as various or as diverse as small molecules, antimicrobials, osmotic changes, or even temperature. And because there are so many different signals, also so many different receptors have evolved. So here are just 4 examples depicted schematically. For example, target aspartate receptors. And all these receptors have a modular domain architecture. So they have various inputs domains. Then they all have a 4-helical coiled transmembrane region. Then they all have a hemp domain and then an output domain. And interestingly, researchers have managed to mix and match these modules, and these receptors still function. And this is what we call chimera. So for example, when we take the output domain of NZ and put it onto the aspartate receptor, we still get a functional receptor. And this is, first of all, very fascinating, but second of all, has the implication that all these receptors must have the same signaling mechanism. But what makes it structurally that they all can have the same signaling mechanism? And this is when you look, or you will see it when you look at the structures of them. And here I show you the AlphaFold 3 models of these receptors. And in red, I highlight the coil-coil backbone. This is the region, a coil-coil region, that goes from the extracellular space across the membrane towards the intracellular space. And this is what they all have in common. And one integral part of this coil-coil backbone is the so-called hemp domain, here depicted in orange. And now we know the structural feature probably that on which the signaling mechanism is based, but the question remains, how is the signal transduced along coil backbone? And thinking about it, one realizes it must be via conformational changes within this backbone. And when one depicts schematically this coil backbone as 4 cylinders, each cylinder representing 1 helix, one can think of several possible conformational changes. One can think of an axial shift, or a rotation mechanism, a scissoring or tilt mechanism, or displacement. And actually, when you go into the literature, you find evidence for all of them. But the most plausible model is actually the rotation model. Let me explain you this diagram and why it's so plausible. So this diagram represents the 4 helices of this coil backbone. And as you might remember from biochemistry classes, maybe, coils are composed of a 7 residue repeat, here indicated as 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7. And in these coil backbones, the core is made up of positions A and D, which are in the sequence position A and 4. And those are hydrophobic residues, and they form the core, and this makes this 4 helical coil stick together. And exactly that conformation is called knobs into holes, and it was expected, of course, for this coil backbone, especially for the hemp domain in this coil backbone. But when the structure was experimentally resolved for the hemp domain, it was found that actually the hemp domain is in a complementary XD8 conformation, which was not expected. In this conformation, 3 residues form the core, the X residue and DA, which is number 1, 4, and 5. And then researchers, they realized that actually if you rotate the helices by 26 degrees, you can interconvert these 2 conformations. And this led to the proposed model that this is the rotational model that leads to the signaling. One is the on state and the other one the off state. However, one has to keep in mind, all these studies have been done on fragments of receptors, based on mutants of receptors, but nobody to date has seen a full-length receptor at work. And thus, in our lab, we have established a model receptor which is called AF1503. This receptor is from Archaeoglobus fulgidus. It is rather small. It has only 338 residues per monomer. My colleagues have experimentally determined its structure. And when they did so, they always found palmitic acid in the PAS domain, so in the sensing domain. So we think of palmitic acid as the ligand for this receptor. My colleagues have managed to build functional chimeras of this model protein, which indicates that it has also the same signaling mechanism, and then that we can extrapolate from this model receptor to the whole 2-component signal transduction receptor family. In my lab, we run biochemical experiments on this receptor and NMR experiments. But on my side, I use the molecular dynamic simulations to study the conformational changes within this receptor. And I think when you do molecular dynamic simulations, it's always important to do a stepwise approach, to go from small to big, because this really ensures that you get biological plausible results. So I started with simulations of the hemp domain and then continued with simulations of the full-length receptor. So let me show you the results for the MD simulations of the hemp domain. So the idea was, okay, we have this rotational model for which 2 conformations are proposed. So the question is, can we sample them? So I set up the simulation. I used the structure of the hemp domain. I performed very general preprocessing of a protein in solution using Chromax and the CHARM 60M force field, of course, water, ions, and regular energy minimization and equilibration scripts. I performed production runs of 1 microsecond simulation with multiple replica and all atom simulations. Of course, after the simulation, I performed sanity checks to see is my protein stable. And then I determined the rotational state along the simulation. How do we do this? So along the simulation, we extract a structure from the simulation of every 100 picoseconds. And then for every structure, we calculate the rotational value it has. And this we can do because coils can be parameterized. And then we have a tool called SAMCC, which exactly gives 1 value for a structure of 4 helical coils that describes the rotational state. If it is 0 degrees, we know it is in the knobs into holes conformation. so the canonical conformation. If it is below 0 degrees, like at around minus 15 degrees, we know it's in XDA conformation. So when I ran the simulation, I found first good indication the hemp dimer is stable in simulation. As you can see from this figure, it's the hemp domain color-coded by the root mean square fluctuation. And then, of course, I looked at the rotational state of the hemp dimer, and we saw that the hemp dimer is preferably in the XDA conformation, as we see that the rotational values are mostly within like minus 10, minus 12 degrees. We did saw some, let's say, outliers of the rotational values towards knobs into holes, so towards 0 degrees rotational state. I can show you one small example here, where at around 220 nanoseconds, the rotational value increases towards 0 and drops again. So it did visit the knobs into holes conformation shortly, but it wasn't stable. It didn't want to stay there. So I thought, maybe if we increase temperature, we would see knobs into holes conformation more often. And I did so, and this distribution stayed similar. And same even when we went up to 90 degrees Celsius. So the dimer was super stable, and it really liked the XDA conformation. And then I also tested a mutant, which is known to be less signaling competent, for which we think it might be in the other conformation. And we see a shift in the distribution, a shift towards knobs into holes, but still the distribution is mainly in the XDA conformation. So what can we conclude from this? We can see our unimodal distribution. So we do not sample both two conformations. The hemp prefers one of the conformations, the XDA conformation. Sometimes we see it shortly rotating into the knobs into holes conformation, but it's not stable. These results are compatible with the rotation model, but one has to keep in mind that this is what we looked at as the hemp in isolation. And normally the hemp domain is part of a whole coil backbone. So the situation might be completely different when we look at it in a full receptor. And this is what I did in the next step. So I looked at the full length A1503 with MD simulations. And in particular, I wanted to know the effect of the ligand, palmitic acid, on the conformation of A1503 and see if I simulate it once without and once with, if I can find two different conformations of the hemp domain. So I set up the simulation using A1503 without and with palmitic acid, performing pre-processing, and this time I embedded the protein in a POPC membrane using the CHARM GUI and a little bit more extensive equilibration scripts. Performed the production runs, each 1 microsecond, 3 replicas. Then I performed a sanity check and then determined the rotational state as you have seen before. And here again, we can see that our protein is stable in simulation without palmitic acid and with palmitic acid. These structures are again color-coded by root mean square fluctuation. What is also obvious from these pictures is that without palmitic acid, the binding site for palmitic acid becomes more flexible. And palmitic acid actually stabilizes this binding site, here indicated by the lower RMSF, root mean square fluctuation of the near residues. But then, of course, the question was, do we see a difference in rotation? And here I plot the rotational value for the time of the simulation of 1000 nanoseconds, in red of A1503 without palmitic acid, and in blue 3 replicas of A1503 with palmitic acid. And what you see is no difference. So we don't see any difference. The hemp domain stays in the XDA conformation. No, there could be different potential explanations. So either the simulations are too short, or there's a too high of an energy barrier. And this is why in the next step, I plan to use enhanced sampling methods, especially the Gaussian accelerated MD. The other reason could be potential explanation that palmitic acid is not the ligand or not the only ligand. How do I come up with this potential explanation? And this arises from an observation of homologous proteins. So we looked for homologous proteins of A1503, and we found 2 very interesting proteins. The one protein here on the left is a protein without an extracellular domain. So the question is, where's the signal come? Like, where does this protein detect a signal? And we think that maybe here in the membrane, there's another protein that detects the signal and then relays it to this protein. And this hypothesis was also supported by another homolog that we found that actually has an N-terminal transmembrane domain. And here we think the signal might be detected by this, then relate to the receptor and further causing downstream signaling. So this has implications for A1503 of maybe the signal does not or not only come from the pus domain, but maybe from a different protein that we don't know yet that is in the membrane. So future work will definitely include homology searches and interaction predictions. So to wrap up, we've seen today that two-component signal transduction systems are at the core of prokaryotic signal transduction. These receptors are dimeric with a coil-coil backbone that I have depicted in this red color. We use the model receptor A1503 to study the signaling in this kind of receptors. There's a proposed rotational model for signal transduction, but so far in our simulations, we couldn't see this in our simulations. We saw that the HAM domain shows a preference for the XDA conformation. And in the MD simulation of the full length A1503, we didn't see an effect on ligand binding. So future work will definitely include enhanced sampling methods to be able to maybe cross energy barriers and to search for additional signal input. Maybe we find a missing piece for the story. And with this, I would like to thank especially Andre, my supervisor, and my colleagues in the Department of Protein Evolution here in Tübingen. And I'm happy to discuss more later. Thank you.
Perfect. Okay. Hello, everyone. Good afternoon. I am Zuzana Janáčková from IUCB Prague, and I would like to take you for an interesting way towards the penetration mechanism of arginine peptides. Why should we talk about arginine peptides in the first place? Well, they are able to passively translocate across the cellular membranes, and they also are able to carry some cargo with them, which can have many applications, such as targeted drug delivery. But the drawback of this system is that are not very well studied yet, respectively. There are two mechanisms how they can get into the cells. One of them is active, that's endocytosis, and that one is very well studied. But the passive mechanism, which does not include endocytosis and any other transporters, that one is largely unknown. So how do the peptides actually penetrate? Well, that is an interesting question because arginine is positively charged while lipids are non-polar. And the other interesting fact is that there are differences between penetration of arginine peptides and lysine peptides. Why is it so interesting? Because arginine and lysine are generally very similar amino acids. They are both positively charged. They have both one positive charge, but there is a difference in the charged group. For arginine, it's guanidinium, whereas for lysine, it's just an amino group. And the guanidinium group has some interesting properties because the guanidinium ions can perform this stacking, which is similar, like, for example, with the pi-pi stacking of benzenes, because they are partially delocalized. So you can see in this picture that they actually aggregate, which can make some differences between the penetration of arginines and lysines. In our group, there was proposed a penetration mechanism which contains the R9 absorption on the membrane, then the curvature and bifurcation of the membrane, cross-linking of the two membranes by arginine peptides, then zipping the membranes together, creation of a fusion stalk, and at the end, finally, the pore formation and penetration of arginine peptides into the cell. But we don't have any direct data that would support this mechanism. So we are working on it with many different people in our group. we actually have a cryo-electron microscopy data of large unilamellar vesicles from Mattia Morandi, and they show us that LUVs without arginine peptides are basically normally like unilamellar and without any weird things on them. But if we add R9 peptides, they are very different. They start to have this weird carved features on them, which resemble as these bifurcations or curvatures that we propose in the mechanism. And they can also create these multilamellar structures, which reminds us cross-linking or zipping, and maybe even something that looks like a fusion. According to this data and this mechanism, I will talk about 3 different projects that are connected together. And the 3 questions that we ask are if R9 peptides influence curvature, if they influence zipping of the membranes together, and if they influence membrane fusion. I will start with the curvature project. And I will show you the systems which we studied. We decided that we will study 2 systems, one with curved membrane and one with almost flat membrane. We decided to study the membranes in this state of bicells because it's much cheaper, because the system can be smaller if we use these bicells, and already the system is pretty big. So to save some computational resources, we decided that we will study these bicells. And for the composition, we used a similar composition that was used for some previous experiments. So we used DOPE, DOPC, DOPS, while DOPS is actually negatively charged. So the membranes which we used were slightly negatively charged. As a collective coordinate, in this case, we used the number of contacts between membrane and peptide heavy atoms. And we used 1 nona-arginine, respectively 1 nona-lysine peptide. That was chosen as a control because that one should not bind that much. From the methods perspective, we used umbrella sampling combined with replica exchange, because without the replica exchange, we were not able to reach the convergence. And if you are interested in the experimental details, you can ask later, but I will rather show you now the results. So these are the 3 free energy profiles depending on the number of contacts between the membrane and the peptide. The blue one is the K9 peptide with a curved membrane, and you can see that there is very minimal interaction. The curve almost doesn't go below 0. So this tells us that K9 peptides almost doesn't interact even with the curved membrane. The orange one is R9 with the flat membrane, which there is interaction, but it's weaker. Whereas for the concave membrane, the interaction with R9 peptide is much stronger. This can tell us that R9 peptides prefer the curved membrane, and this actually makes sense already from the very basic perspective that when the peptide like the membrane, then if the membrane is curved, there is more space for the interaction. So it makes sense that we have these results. And when the peptides prefer the curved membrane, it can also mean that the R9 peptides might cause some type of membrane curvature and some structures that we see in the mechanism. So this was the curvature project, and now let's talk about the zipping. For the zipping, we decided to use periodic membranes with the same composition. And the systems, to be able to show the zipping and the movement of the membranes, we needed to use water pores. So each membrane has water pores that enable diffusion of water and ions. And we decided to use one system without peptides and one with 5 non-arginine peptides. We expect, respectively, we already showed that if there are no peptides, there is no zipping. The membranes just stay approximately in the same distance as they were at the very beginning. With the arginine peptides, we are trying to look for some method that could show us if there is some zipping. The technical challenge is that the system is really big, and it takes a lot of time to equilibrate every single small change. So we did not find any method working yet, but we are working on it currently right now. The other thing that we are working on right now is trying to show this in experiments. We started a collaboration with Raya Sorkin from University in Tel Aviv, and we want to do experiments via optical tweezers. This experimental setup uses membrane-coated beads that are held by lasers. The basic mechanism is just that there is different refraction index of the bead and of the buffer that allows us manipulate by lasers with the beads. And it also enables us to measure very precisely the force which is necessary to manipulate with the beads. So it can show us if there is some interaction between the membrane-coated beads with and without arginine peptides. And again, we expect no interaction without peptides and some interaction with non-arginine peptides. So the membrane zipping project is now the one we work on. And the other one, which we also work on currently right now, is the fusion project. For this project, we use memfusion collective variable, which is implemented in Plumet, and it basically allows us to drag the ends of the lipids that are depicted in red in this picture to the center of a cylinder which has specific layers. Mathematical implementation of this collective coordinate is a bit complicated, but you can see in the video how does it look like when we apply this collective coordinate. It basically drags the lipids to the center and creates fusion stock. We used umbrella sampling again, and we used one system with curved membrane and one system with flat membrane. Why these two systems? Well, you saw in the mechanism that the curvature plays an important role in the whole process. So we wanted to try to figure out if the curvature plays a role for the fusion. We again use the same membrane composition. And for now, for the simulations that I'll show you, we did not use any peptides. We will add the data with peptides later. And we already have some work in progress data of not completely equilibrated simulations, but you can already see that there is some trend. The flat membrane to form the stock has much higher free energy barrier than the curved membrane, which means that the curved membrane to fuse needs less energy and we can see some influence of the curvature on the formation of stalk. This is also a project we are working on right now. So I would like to wrap up the whole presentation. Using the data you see, and we are also currently working on right now, we can more closely examine how curvature, zipping, and membrane fusion work. And together with experimental results that other people from our group work on, we can better understand the passive penetration process, which hopefully at the end will help us create better cell-penetrating peptides for drug delivery. At the end, I would like to thank to my advisors, Semen and Pavel, to my experimental collaborators, Mattia and Raya, and also Katarina and Miguel that helped me a lot to learn how simulations work. And I would also like to thank to whole group of Pavel Jungwirth and my financial resources. Thank you for your attention.
Thank you very much. We hear, Susanna, that you have a public there.
Yes.
Could you stop sharing? Sorry, because we see doubles. Yeah, thank you very much. Okay, so they are not online. They are just physically there.
Yes.
Yeah, okay, that is good. Yeah, so now we don't really have question online, but I will invite everybody online to raise his hand because nobody put a question in a Q&A. So I will invite everybody to raise the hand. If you have a question, we can just unmute you and let you speak it. I think Otto has to habilitate the raise hand option. Is correct, Otto?
Yeah, I have to check if it's disabled. One moment.
Okay, yeah. So thank you all the three. I really enjoy all your presentation. Thank you very much. I think all of three of you has the challenge that you are trying to understand the mechanism is correct. And something that probably you didn't decide it. It's not a mechanism that you plan. Someone else planned it, the nature plan most of the time. And it's nice to see that MD now, molecular dynamics, can not only predict the structure, but can also provide us information of mechanism. Do you see, I have a general question for all of you, meantime people raise the hand. Where do you see the challenge to address and to answer this question, to understand what is behind, for example, what triggered the catalysis, what is the mechanism behind all the things that you have shown using molecular simulation? Do you see any challenge in that?
I personally still think challenge is within the force field. As I mean, I think there's a lot of discussion on this. They're still not optimal and still approximations
Yeah.
Of the reality, and not falling into the trap of blindly using simulations and force fields and believing in what you see, and really double check everything and make sure you actually get biological results, then maybe validate them experimentally. But it's just also not only the challenge, but then also the possibility of it then, if you have a hypothesis, to then experimentally validate it.
Yeah. So I have a following question, and then I ask also to the others. So I was curious because I think you use AlphaFold to predict your structure in AlphaFold 3, if I was correct. How you do? Yeah, so it's correct. Yeah. So how do you see, because AlphaFold somehow is also prediction, like also force field are prediction from completely different point of view, but they are both prediction. How is could you comment what would be, one has a training set that has which is trained, and then it provides you an answer. And the other is based on some decision that we have made on how describe physical interaction with particle. And also there we have different procedure, also sort of training, because we parameterize against different type of data. So, but they are on behind the algorithms is very different. So do you feel that also what you were mentioning about force field, it will be also a challenge for everything that comes from a training, AI training like AlphaFold, or you think is in a different way?
So first of all, the protein I use for simulation, we have experimental data for, but the other proteins, this were AlphaFold predictions. Yeah, I think it's important to think about the difference between physics-based methods and AI-based methods. And as far as I'm informed, AI-based or machine learning-based force fields are also on the rise. I haven't used them. I'm also still skeptical about them. And I think it's great that we have such a good physical description of the intramolecular forces and everything that I think AI-based methods for simulations won't replace physics-based methods completely, but maybe they will complement each other. And for AlphaFold structure prediction, I do believe that they are now really good and very confident, except for some special cases. For example, I've seen for very long receptors, or any protein that is very long and fibrous, AlphaFold makes just super crappy predictions because it kind of folds the protein on itself at some point. And you still have to still look at the proteins and make a educated decision about whether the prediction makes sense or not. And I think the same applies for MD simulations, in my opinion.
Yeah, thank you very much. I saw that in the meantime, it popped up a question. Matteo, I don't know if we can speak. Could we unmute Matteo as a question for Susanna?
Let's see.
Yeah, Matteo, could
All
you
right.
speak?
Yeah.
Yeah,
Can you hear
please
me?
go ahead. Yes, we hear you
Yes.
very well.
Okay. So, first of all, okay, thank you for all the presentation. Thank you, Susanna, for your presentation. Okay, what I asked was, I will read again. My question is, how about the zipping mechanism you described? How those water pores are generated, and if they have some physical or biological meaning?
Thank you, Mata, for your question. They don't have any biological meaning. They are there really only so we can see the moving of the membranes in the Z direction, and they are implemented. Well, actually, we started with the pores that were implemented in Plumet, but now we use pure Gromax. So we just use some artificial potential which creates these pores. I think we use a potential that is applied only on the lipid tails, and it kind of restricts the tails from going to the center with some diameter around it. So it's basically just very basic artificial potential that creates these pores. Did I answer the question?
Yeah. Yeah. Thank you very much, Susanna.
Actually, I would also have a question for Jan, if it's possible.
Yeah, go ahead, please.
So Jan, I was wondering, for this mechanism, how does it depend on the DNA sequence, or is it DNA sequence independent?
Yes, well, this is one of the things that would be very interesting, I guess, to study. Currently, this is just some symmetrical or palindromic DNA segment which we use. But presumably there is some dependence on the sequence because, well, the topoisomerases need to somehow know where to bind. But yeah, maybe answering this with through molecular simulations is not the best approach, because you would need to run very many different simulations, and presumably this can be done easier through experimental methods.
Yeah, thank you.
Yeah, thanks for the question.
So we still have some minutes I think we don't have anybody that want to have but I will go on. So, Jan, Susanna, what do you think on the challenge that you see in the MD now that you have just tried, in particular to address a mechanism?
Yeah, I guess I can answer that
Yeah,
on
go ahead.
my case specifically. So I showed that we look at the mechanism where there is some bond breaking and some so ATP hydrolysis. And well, I guess the very obvious limitation of MD is that whatever we start with is what we have to finish with. There is no bond breaking. So if we want to somehow simulate this system, we need to use very many different starting structures and then piece it together piece by piece. And additionally, since we are simulating very large, I think all of us are simulating very large systems, obviously there's always, at least currently, going to be the timescale problem. So we need to use some kind of enhanced sampling methods which skew reality to get some answers to our questions.
Yeah, probably what you also aim to, what in your answer is that you would like to have an approach that scale between also models. So
Yeah.
from a classical model to a quantum chemistry model base, for example, that will
Yes, exactly.
solve something on your problem, but you don't want that the other part, the quantum chemistry part, is slowing down because you have a large system. So you want to access a long time scale. So that is indeed the challenge, how to bridge all different time scale. And and also model. Yeah, indeed, that I think is one of the big challenges we are also together. I agree also with the force field, how we model things. And Susanna, please, do you have something to add?
Maybe from my perspective.
Yeah, sure. You should be from your perspective.
Yeah, I'm not sure if it applies also on the other projects, but definitely for me, in the in our attempts to resolve some big mechanism, the challenge is that the system that we want to study is too big to study with MD. We would really need millions of atoms. Possibly we would like to study this the whole cell, which is absolutely impossible. And the other problem is that for experimental study, the system is too small. So we kind of need to
I think we lost Susanna.
work on
Okay.
some of them try to resolve experimentally, some of them by simulations. But we somehow need to find the connections between these different techniques so we can somehow sew it together and look if we find some compatible results.
Yeah, so somehow to bridge a system, an experimental data that is done on a larger system, on a system that you have a final size. So then in some sense, you cannot expand as as large as you would like, because you could have size effect. Yeah, that I think in particular in your cases might be a challenge. Yeah, yeah. But maybe in the future you could access cell. At least there is some starting in minimal cell. There was a presentation with Martini force field. They start to simulate a minimal cell. So maybe. I don't think atomistic. We are still a couple of sometimes to go, but yeah, at least we are. We were not thinking that was possible, I think 5 years ago. So then maybe it's positive. Yeah.
Yeah, I mean, if we could simulate the whole cell atomistically for 15 minutes, then I think we would be done. But I don't think this will happen.
Not now. Okay, thank you all of you for the nice discussion, and for the public to attending the webinar. Thank you very much to all of you. We now close this section. Thank you very much. Bye-bye.
Thank you. Bye.